So, low-calorie sweeteners, why is there a debate? Well, the answer basically is that low-calorie sweeteners will always generate lots of interest, for good or bad, and there's no more trendy topic than the gut microbiota at the moment. So that's, in a nutshell, that's why there's a debate. In order to talk about low-calorie sweeteners and the gut microbiota, I need to do a fair amount of introduction to the gut microbiota itself and what factors influence it, and the impact on host health, and particularly looking at the microbiota and links with diabetes and metabolic syndrome. So I hope you'll bear with me, because in order to understand or put into context the impact of LCS on microbiota, you need to know a bit more about the microbiota. So first of all, lots of large things associated with human microbiota, even though they're small organisms. So there's a very large number of organisms there, around 40 trillion organisms associated with a human gut, and it's approximately the same number, about 50-50 human cells to bacterial cells, and importantly, many have not been cultured yet. In addition to there being a large number, there's a huge diversity of organisms there. Around 1,000 or so different bacteria, archaea, types of organisms have been identified, and about, in any one individual, about 150 would predominate. In addition to the organisms, there are around 3 million genes. That's 150 times bigger. The gut microbiome is about 150 times the size of the human genome. About a third of these bacterial types are common to most people, and very importantly, there is a large inter-individual variation in the types of bacteria you have in your gut. It also has a large metabolic potential. It's considered to be equivalent to the liver, but is very different from that of the liver. It's more reductive and hydrolytic, where the liver is oxidative and conjugative, but very big metabolic capacity. And the other important factor to consider is that the distribution of the organisms in the gut is very uneven. We have very few organisms in the stomach because of the acidic conditions of the gastric juice, and in the small intestine because the peristalsis keeps moving the food through and they can't establish a microbiota there. So the colon is the main site of the microbiota. So in order to have an impact on the microbiota, if you take a dietary compound, it has to reach the colon or somehow influence the colonic environment in an indirect way. And that's very important from the point of view of low-calorie sweeteners. Now, in terms of how we investigate the gut microbiota, the old-fashioned method, which I used to use a long time ago, was a cultural method. You plated fecal samples out onto Petri dishes. But we know now that you only can cultivate about 10 to 30% of the gut microbiota. Molecular methods are now the norm for investigating the microbiota. They speed up the analysis considerably, and you can do gene sequence analysis of a section of the DNA, which is called the 16S rRNA, which is present in all microbes, but it's very variable, so you can identify organisms that way. Or you can do metagenomic sequencing, where you take a total DNA sample and shear it by shotgun approach, and then sequence the genes. What's important here is that the methodologies yield different results. And even the same methodology done in different labs can yield different results depending on how they do the study. So it's quite difficult at the moment to compare studies of microbiota. So this is just a few of the organisms. I've said about 1,000 different sorts. These are the main ones. They fall into two main phyla. So 90% of the bacteria in your gut fall into what's called the Firmicutes or the Bacteroidetes phyla. So Firmicutes include things like Clostridia, you've probably heard of, Lactobacillus, Streptococcus, Butyrvibrio, which produces butyrate. The Bacteroides, two main groups of Bacteroides and Prevotella, but there are also other interesting ones. Akkermansia is quite an interesting organism. There's a lot of work now looking at its association with a health benefit in terms of cardiovascular disease. And you've got Escherichia coli, and Bifidobacteria, often used as probiotics, come under the Actinobacterium. So there's just a few examples, and there's obviously a lot more. A load of factors affect the microbiota. your general lifestyle, where you live, in different parts of the world, people have different microbiota. Host genetics has an impact. Early colonization, whether it's a cesarean delivery or vaginal delivery, can have a big impact on the microbiota, and early breast milk versus formula milk. Drugs, antibiotics, obviously can destroy the microbiota, and various lots of different health factors and disease can have an impact on the microbiota. But also very important is diet. So the dietary pattern has an impact. Western diets, vegetarian diets, specific types of foods, so whole grain diets, particularly vegetables, fruits and vegetables, can have an impact. Fiber has an important impact on the gut, but fat, protein, phytochemicals are a very interesting area now, and also these food-associated commensals, probiotics, lactobacilli and bifidobacteria. In the light of this, it's important to control a diet when you're conducting any human studies on the microbiota. You need to control the diet of the people that you're looking at, or assess the diet very, very carefully. And if you don't do that, then all these different factors can have an impact on the outcome and confound your results. The other important point to mention is differences between human and rodent microbiotas. They're both dominated by these Bacteroidetes and Firmicutes phyla, but there are big differences in the types of bacteria you get in the different in humans and mice. So 85% of the genera of organisms in the mouse microbiota are not present in humans. So in humans you get Prevotella, Faecalibacterium in mouse, Lactobacillus is very important, Alstipes and Turicibacter. The other very important thing is that the distribution of the microbiota is different in rats and mice by comparison to humans. Mice don't have such a acidic gastric juice. So in the mouse stomach and the small intestine, there is a fairly stable microbiota. In the human, small intestine and stomach have very few microbes. So there's more opportunity for substances which are relatively easily absorbed to be affected by the microbiota in mice. But the takeaway point from this is that extrapolating results from mice and rats with regard to microbiota and towards human health is not very straightforward. We know that the microbiota has a big impact on human physiology. It has an impact on the immune system, a very big impact on the immune system, on digestive function and metabolism of dietary components and foreign compounds and drugs. And there's also a lot of evidence that the microbiota is associated with disorders, and particularly gut disorders like gastroenteritis, inflammatory bowel disease, irritable bowel syndrome, and colorectal cancer. And we know this because you can show there's disease instances and severity is different between germ-free animals and animals with a microbiota, and also a load of studies looking at the microbiota and diversity of the microbiota in patients with a disease and healthy subjects. And obviously a lot of the interest now has been generated by studies which suggest that the microbiota may be involved in obesity and in type 2 diabetes and metabolic syndrome. Just briefly, can we define a healthy microbiota? And I don't think we can, because there's such a wide range of inter-individual differences in people's microbiotas. The second question is, can we define a non-healthy microbiota? Now we know, as I've just mentioned, there are differences in the diversity and the types of bacteria you get in various diseases. And this has led to the concept of dysbiosis. That is, in diseases, the microbiota can be aberrant, it can be disturbed. But as you'll see in a moment, there's a lack of consistency between studies, although there's some consensus that low diversity of types of bacteria may be associated with poorer health. And there's the big question is this dysbiosis, is this derangement of the microbiota a cause or a consequence of a disturbed gut environment? For example, if you've got inflammatory bowel disease or colon cancer, there's a lot of blood going into the colon, and that's going to have a big impact on the types of bacteria will utilize anything they can get their hands on. So it's quite likely that your microbiota is going to be disturbed under those conditions. And at the moment, there's no way you can use dysbiosis or disturbed microbiota to diagnose or predict the onset of a metabolic disease, such as obesity and type 2 diabetes. So I just wanted to have just briefly talk about what we know about the microbiota and obesity. A load of studies looking at obese people versus lean people, or looking at weight loss diets or surgery, indicate that these two main phyla, Bacteroidetes and Firmicutes, are related to body weight regulation. But of course, that covers 90% of the bacteria there. But the studies are very inconsistent. The early studies showed that there was a decrease in Bacteroidetes in obese, often with an increase in Firmicutes. So you had this low Bacteroides-Firmicute ratio. But then there was an equal number of studies which showed no association, or in fact the opposite trend. And if you drill down into the types of bacteria, different studies have shown different effects. There is, as I mentioned earlier, some evidence that a low level of this organism called Akkermansia may be associated with obesity, but it's not very convincing. And these differences in outcome from these different studies is probably related to the analytical methodology that's used, dietary control, the size of the study, and so on. And a number of these references I've given here they summarize the statements they make. They would say, due to the complexity of the species associated with these main phyla and the individual differences in microbiota between people, it's difficult to interpret any overall change and to establish precise relationships between obesity and the presence or absence of a specific group. Similar pattern with gut microbiota and type 2 diabetes and metabolic syndrome. There have been 4 studies looking at the microbiota associated with type 2 diabetes, 1 study with metabolic syndrome and comparing it to healthy people. The studies are really quite inconsistent with regard to the presence or numbers of specific microbial types. There's a bit of evidence that suggests that low levels of an organism called Faecalibacterium prausnitzii, all these organisms, they're very small, but they have huge names usually. But Faecalibacterium prausnitzii might be important, and there's some evidence this is a sort of anti-inflammatory organism. And also that butyrate producers are associated with type 2 diabetes and metabolic, low levels of butyrate producers associated with metabolic syndrome. And there's some studies in mice that provide evidence for causal links, but we really need more evidence to establish whether the associations that we've seen already are causal and try and identify the main organisms in humans. We don't know much about the mechanisms of why the microbiota might influence any of these diseases, but short-chain fatty acids, SCFA, come up very often. These are products of bacterial fermentation of non-digestible dietary carbohydrates like dietary fiber and oligosaccharides and resistant starch. And they generate 3 main short-chain fatty acids: acetate, which can be used as an energy source, butyrate, which is an energy for colonocytes and can influence GLP-1, and propionate, but also increases these hormones, these gut hormones, PYY and GLP-1. But short-chain fatty acids, although they are probably involved, we don't know how, because they have very diverse effects on, for example, energy metabolism, both positive and negative. They can enable energy to be recovered from dietary fiber, so you might expect high levels of short-chain fatty acids might be associated with obesity. But on the other hand, they can increase satiety hormones and suppress intake of food. And they also, there's some evidence they increase insulin secretion and sensitivity. So finally, I get on to what do low-calorie sweeteners and the gut microbiota. How do they interact? Just a few general considerations. Low-calorie sweeteners are very diverse in structure, as we know. Their disposition in the body is very variable as well, so it's unlikely that they'll all have similar effects on the microbiota because they're all different compounds. But we know that some, like aspartame and acesulfame K, are completely metabolized and absorbed in the small intestine, so they won't reach the colonic microbiota. Therefore, direct effects on human microbiota is unlikely. It might have effects in mice and rats because there's a microbiota in the small intestine. And others, like sucralose, are not absorbed, but they're not metabolized either by bacteria, so they're unlikely that the bacteria can use them as an energy substrate. And as we've heard earlier, studies often use very high doses, and a characteristic of a lot of the studies that I'm going to talk about are that they have very poor dietary control. So here's a summary of all the sweeteners and all the studies on one slide. This is the types of studies that have been done. So saccharin's they've been looked at in rats, mice, pigs, and humans, and changes have been seen. I'll go on to those in a moment. Sucralose, in studies in rodents, changes have been seen, but their significance is questionable. Aspartame, changes seen in rats. Mice and human cross-sectional studies don't show very much effect at all. Sucram, there's a study in pigs which showed an increase in lactobacilli. Aspartame K, inconsistent changes in mice. There was a human cross-sectional study which showed small changes in diversity of the bacteria, but they're very small. Cyclamate in monkeys, no effects. Neotame, decrease in the firmicutes bacteroides ratio and some metabolites. And then steviol glycosides, a study in mice which had no major effects, and really no human studies apart from one just looking at effects in vitro using fecal samples and seeing whether if you add steviol to the fecal sample you've got an effect. But anyway, minor changes. So I just want to go into one or two of those. One or two of those in more detail. First of all, saccharin. Saccharin has been looked at a lot in terms of microbiota. Some early studies, which I was involved in, and others gave very high doses, to my shame, very high doses, and saw an increase in fecal bacteria, decrease in enzymes, especially short-chain fatty acids. There was an increase in what looked like bacterial protein metabolism in the animals, but this was not reproduced when the study was done in humans. A recent study in mice at very low doses in drinking water showed increased in inflammatory markers and an increase in 6 types of bacteria: Akkermansia, Roseburia, Turicibacter. And I've just put that might be beneficial. Akkermansia we think is quite beneficial. Roseburia is a butyrate producer, which might be beneficial. We don't really know, but certainly I wouldn't say that was an adverse microbiota. Decrease in 4 genera, and there were some increases in pro-inflammatory mediators from the gut. Whoops, gone the wrong way. Then this is the Suez, the notorious or famous Suez study, depending on which side of the bridge you are, who looked at a series of studies in mice and humans. So he gave mice saccharin in the drinking water at very high dose versus control for 11 weeks. There was increase in Bacteroides and some Clostridia, decrease in other Clostridia and Lactobacillus. There was an increase in fecal short-chain fatty acids. This is very odd. He found no short-chain fatty acids in the control group, and then it went up to 0.8 or no acetate, rather 0.8 of a millimolar of acetate. This is really bizarre, and obviously I think his methods were wrong somehow. And a huge amount of propionate by comparison to acetate. This is never seen that before. But importantly, food consumption was 50% lower in the saccharin group. So that kind of means all the other the kind of things which we know influence the microbiota are going to be affected by this lower incidence. He did see, though, changes associated with glucose intolerance and was able to transfer the microbiota to germ-free mice, which indicates there is a possible role for the microbiota. In the human studies he did a cross-sectional study with 172 high and low consumers of a variety of low-calorie sweeteners, saw increases in various types, Enterobacter, Proteobacter. There was no dietary assessment, and again, no control. And then he did a study in 7 subjects, given saccharin, again, quite a reasonable dose for 5 days. Half the subjects showed poorer glycemic response, which we've just heard about, and there were changes in microbiota, increase in Bacteroides, decrease in some Clostridia, increase in Lactobacilli. The changes he saw were different to those in the observational study. And here there was no untreated control group, no dietary control. So it's very difficult to conclude anything from that study. Also, a lot of studies been done with sucralose. There was a study, an older study in 2008, looking at, this used Splenda, so it looks a high dose, but actually it was Splenda, which is only a fraction of Splenda is actually sucralose. And they saw small differences, lower than log 10, which is very, very minor in total anaerobes, and some changes in some of these things. And they were measured by viable counts, so they're probably missing a lot of organisms. A study in mice showed decreases in Streptococcus, Staphylococcus, had an increase in Akkermansia and an increase in Rosbura. Again, could be I'd have thought that might be a beneficial change rather than an adverse change. But there were changes in fecal metabolites and some inflammation. This is a study in sucralose, 1.5 to 15 mg per kg per day, so quite a high dose. No major changes in this study. There was an increase in butyrate, and there was no effect on weight or fat levels. And then a recent study, sucralose again, at a reasonably low dose. Firmicutes were increased, Bacteroides decreased, Bifidobacterium increased. So really quite a wide range of results on microbiota, no great consistency at all. Aspartame and the microbiota, two main studies. This is obese rats fed a high fat diet plus or minus aspartame. They found increases in Clostridia strains, Rosburia, Enterobacteria, a decrease in Bifidobacterium, and a decrease in this Firmicutes Bacteroides ratio. There was an increase in serum propionate, short-chain fatty acids, decrease in weight gain, and increase in fasting glucose. But these results are confounded by large differences in other factors. So the food intake was lower by 25% in the aspartame group. The body weight was 17% decreased, and the water intake was 40% lower. So again, all of these changes could be due to these changes in diet. There's a human cross-sectional study of aspartame consumers, quite a wide range of intakes, really no difference in abundance, in the Firmicute ratio or microbial function changes. A few changes in diversity, but there's only 7 people in this study. So the key conclusions are there are differences in human gut microbiota associated with health, but the evidence for a causal link with metabolic syndrome and obesity, I think, are very limited at the moment. The bacterial types and metabolites and the mechanisms underlying this host microbe interactions in health are not fully elucidated. Most studies of low-calorie sweeteners on the microbiota are in rodents, and the effects are relatively small, and they're confounded by inadequate control groups and the lack of any dietary control, usually. There are unfortunately very few human studies on low-calorie sweeteners and microbiota, and in general, the effects are inconsistent. They're very difficult to interpret due to lack of dietary control or assessment, and the lack of any control groups. So really we need well-controlled human studies if we want to explore further this interaction of low-calorie sweeteners with the microbiota. And I think the studies that need to be done should be looking at the functions of the gut bacteria rather than just what types of bacteria are there. And my overall conclusion would be that the current evidence does not support that low-calorie sweeteners have an adverse effect on health via impact on the gut microbiota. So thank you for your attention.